Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So we were discussing about neurobiology, in which we have already covered the structural aspect of neurons, and then we also discussed the neuroglial cells, and in last class we discussed the myelination of the axons. In today's class, we are going to discuss about ion channels. So, for the purpose of understanding the electrical signaling, the organized signaling that happens in nerve cells, how the nerve impulses are conducted from the dendritic part to the axon terminals, we first need to know about ion channels that are present on the membrane of neurons. So, we already know that neurons are the excitable cells, and the excitability is due to the channels which are present on the membrane, which contributes into the uneven distribution of ions in the extracellular matrix and the cytosol of the neuron, and that makes the neurons excitable. So whenever a stimulus arrives, the membrane potential of the neurons is changed, and that is why neurons, those typical cells, are the excitable cells. Muscle cells are also excitable cells. So in today's lecture, we are going to discuss about the ion channels that are present, the different types of ion channels that are present on the membrane of neurons. So we already know that the channels are the protein molecules, protein moieties that are present on the membrane, which helps in the exchange of a molecule from either the extracellular matrix to cytosol or vice versa. So we already know that on the membrane there are the channels. So this is a channel, okay, and there will be our molecule, let's say water molecules, okay. There will be distribution of water molecules on the extracellular matrix as well as in the cytosol, okay, and then it will set up certain concentration gradient. Let's say that water molecules are more on the extracellular matrix, so water concentration is higher in the extracellular matrix. That is so the movement of the water molecules will be according to the concentration gradients. In the cytosol, water molecules are less compared to the extracellular matrix, so this sets up a concentration gradient from the extracellular matrix to the cytosol, and that is why the water molecules will move from the extracellular matrix to cytosol. So the molecules move across the membrane through channels down the concentration gradient. Okay, so we already know about this. So in the case of ions, in the case of neurons, ions move across the membrane of neurons through channels down the electrochemical gradient. Okay, so we already know about the concentration gradient. So, but in the case of ions, the electrochemical gradient is considered, means this has two aspects. First, electrical aspect, and the other one is the chemical aspect. The chemical driving force is the concentration gradient. So wherever the concentration of ions is high, from the highly concentrated area to the low concentrated area, the movement will be seen. So let's say this is the membrane of the neuron. So this is the membrane, and there we are looking at the potassium ions. Let's say potassium ions are present on the extracellular membrane as well as in the cytosol. But the concentration of the potassium ions in the cytosol is higher compared to the extracellular matrix. Okay, so this sets up a concentration gradient. So this is the chemical driving force. This is the chemical aspect of the concentration gradient, that the concentration of potassium ions is high inside, and that is why the flow of the potassium ions will be from inside to outside. Okay, so when we look at the chemical aspect, according to the concentration gradient, potassium will flow outside in this particular scenario. Okay, so this is the chemical aspect of the ion exchange, but there is an electrical aspect involved in the ions, because ions are the charged molecules. So whenever we are considering water, glucose, sucrose uptake or transport, those molecules are uncharged, and that is why we do not really consider the electrical aspect. But the ions are the charged moieties, and that is why the movement of ions will depend on the electrical properties as well. So we already know that the positive ions will move towards the negative charge. So in the neurons, we already in the neurons in the last lectures as well, I have discussed this, that the cytosolic part has anions, more anions compared to the extracellular matrix. Okay, and then on the extra, so this sets up the whole total negative charge inside the membrane compared to the outside. So the outer part of the membrane is positively charged, while the inner part of the membrane is negatively charged due to the high concentration of anions on the cytosolic part. So this sets up an electrical gradient, so which forces the positive ions to move from the positive charge to negative charge. So electrical aspect says that the cations will move from positively charged area to highly negative area. So this is relative, okay? So this is not an absolute scenario. This is relative scenario. Wherever there is more positive ions accumulating, the positive, so cations like potassium, cations like sodium, those will move from positive area to the negative area. So while the movement of ions, whenever we take a look at the ion exchange, we consider both of the aspects, the electrical aspect as well as the chemical aspect, and that is why we say that the movement of anions is down the electrochemical gradient. So this is the basics of the so this is the basic that I wanted to clear before discussing about the channels. So those ions they move through the channels, and the types of channels in the neuron membrane, that they are different. Okay, so to which stimulus those channels open, depending on that there are different types of channels present on the membrane. So let's discuss about them. So there are four types of ion channels present on the membrane. First are known as leaky channels or leak channels. The other ones are the ligand-gated channels. Then there are mechanical gated channels, and voltage gated channels. Okay, so those channels, the channel proteins, they have a gate which opens and closes corresponding to some stimuli, and depending on which kind of stimuli, whether it opens upon the arrival of ligand, or whether it opens upon the mechanical stress, or whether it opens upon the change in potential membrane potential, depending on that, these are the different types of channels. Okay, so the leak channels are the ones which open and close randomly. So it doesn't matter whether the stimulus is there or not. The open conformation and close, the interplay between the open conformation and close conformation will be quite random, and that is why they are known as leak channels, because the ions are going to leak through those channels randomly. Okay, so leakage is random, and that is why those channels are called as the leak channels. So there are two types of leak channels: the sodium ion leak channels. Okay, sodium leak channels and potassium leak channels. The concentration of potassium leak channels on the membrane is much higher than the concentration of sodium ion leak channels on the membrane, and that is why we say that the membrane of neurons, it is selectively permeable to the potassium ions. Because naturally, the structurally, the potassium leak channels, they are there will be much more potassium leak channels compared to the sodium leak channels. And again, the leakiness, the membrane usually opens the potassium leak channels, and the opening of the sodium leak channels is quite less frequent compared to that, and that is why we say that the neuronal membrane, it is selectively permeable to potassium ions. Then there is a different type of channels known as ligand-gated channels. So this channels opens the gate upon the arrival of ligand, or let's say the binding of ligand. So once the ligand, for example, the neurotransmitter like acetylcholine, once such ligand binds to the receptor, binds to that channel, the gate of the channel opens, which allows the transport of the sodium or potassium ion channels. So the ligand-gated sodium ion channels, those are the ones which do the influx of sodium ions, means they are going to take sodium ions in the cytosol from the extracellular matrix, while the ligand gated potassium ion channels, they are going to do the efflux of the potassium ions, means they are going to move, the potassium ions will move out from the cytosol, they will go to the extracellular matrix. Okay, so there are two types of ligand gated channels. The ligand gated sodium ion channels will do the influx means the ion movement is from this extracellular matrix to the cytosol, while the potassium ligand gated potassium ion channels will do the efflux, means the potassium ions will be moved, they will be moving out from the cytosol to the extracellular matrix. So those channels, they open upon the binding of the ligand. Okay, then there is other type called mechanical gated ligands. So those ligands, they will change their conformation due to force distortion. Okay, so there will be some mechanical force applied which is going to distort the channel, and due to the distortion of channel, the gate opens, and once the gate is open, the ion exchange will occur. Okay, so this the force distortion would be due to some touch sensation, or due to vibrations, or due to some pressure is applied on the skin. Okay, so touch, vibration, and pressure, though, or maybe there's some stretch on the muscle. So those mechanical, those sensations are sensed, and they exert some force which distorts the channel. Okay, so this is how the ion exchange happens due to the mechanical gated channel. So there is no ligand binding here, and there's no leakiness, like there's no randomness. Whenever there will be sensation of touch, vibration, pressure, or stretch, that force will be distorting the channel, and that will open the channel. So the conformational change is due to the force that is exerted on the specific tissue. And then the last and very important type of channels is voltage gated ion channels. So those ion channels, they open their gate open gate their gate when the membrane potential when the membrane potential changes. Okay, so we have already discussed that in the neuronal cell, in the neuronal cells, the extracellular matrix has higher concentration of sodium ions and chloride ions compared to the cytosol. While on the cytosolic part, there will be the higher concentration of potassium ions and anions. So due to this, the cytosolic part of the membrane has overall negative charge compared to the outer part. So the extracellular part of the membrane has a positive charge, overall positive charge compared to the inner side. Okay, so this membrane potential is about 70 millivolt when the cell is at rest. So when there is slight change in this membrane potential, let's say that this membrane potential from minus 70 goes to minus 55 millivolt, let's say. Okay, why? Because let's say due to some leakage, due to the opening of some leaky channels, the potassium ions move in or the sodium ions move in or move out, and this changes. So here the membrane potential is going from minus 70 to minus 55, means it is getting positive. This means that the positive ions will move in. So let's say that the sodium or let's say here the potassium leak channels open. Okay, and due to opening of potassium leak channels, some potassium ions which are positively charged, they move in. Okay, so this will increase overall positive charge on the inside of membrane, and that is why the membrane potential is changing from minus 70 volt to minus 55 volt. So this change in membrane potential will be sensed by some other channels, okay, which we call as voltage gated channel, and due to this change in membrane potential, they will open their gate, and that will cause the ion exchange. So I hope the types of ion channels is very clear to you all. Let's view them diagrammatically. So here are few schematics which are clearly showing the ion channels that are present in the neuronal cell membrane. So this is the extracellular part of the cell, and this is the intracellular part. So this is the cytosolic part, the yellow one, and the blue one is the extracellular part. And this is the plasma membrane. This is the lipid bilayer where the channels are present. Okay, so these are the channels. These are the different types of channels. So this first one is the leak channel, and this is specifically potassium ion leak channels. So here you can clearly see the potassium ion concentration is very high on the inside of the membrane. Okay, and those leak ion channels, they are going to do the passive transport. Okay, so there's no energy involved, and they are just going to leak potassium ions according to the concentration gradient. And due to the higher concentration of potassium ions on the cytosolic side, the potassium ion will move from the cytosol to the to the extracellular membrane. So these are the normal channels which are going to leak the ions according to the concentration gradient, and they will open randomly. So they do not need any stimulus, they do not need any vibration, touch, or any kind of force for them to open. Okay. Then the second type of channel is the ligand-gated channels. Here you can see on the lipid bilayer, a ligand-gated channel has a very specific conformation, and it is sitting on the lipid bilayer. And here you can see upon arrival of ligand, this red colored one is a ligand. Okay, let me zoom in the picture. Yeah, much better. So this is a ligand. The ligand could be any neurotransmitter like acetylcholine, it could be dopamine, it could be serotonin. So this ligand has so on the channel, on the protein, there will be a site known as ligand binding site. So this is ligand binding site. Okay, that will be present on the channel on which the ligand will bind. So once this ligand binds, it is going to do the conformational change which opens the gate. And once the gate opens, let's say if this is the ligand gated sodium ion channel, so it will selectively pass sodium ions through that. Okay, so the ligand gated ion channels, they are sometimes selective and sometimes they are non-selective. So in this case, this is the ligand gated sodium ion channels. So no, in this case, this is the ligand gated cation channels. Okay, let me write it here. So this is a okay. This is a ligand gated cation channels. Cation channel, which is specific to the acetylcholine, and this is an acetylcholine receptor. So this acetylcholine receptor is the ligand gated cation channel. So this does not so this is not very specific. This is specific to cations. This will not allow the anion exchange, but it can allow any allow the exchange of any cation. So here you can see it is allowing the exchange of calcium, it is allowing the exchange of sodium, and it is allowing the exchange of potassium. So this is potassium, this one is the calcium, and this is sodium. So here you can see the acetylcholine receptor, which is a ligand gated channel. It is ligand gated cation channel. So please remember this information. This will be helpful in future aspects when we will be discussing about synapses, when we'll be discussing about certain acetylcholine receptors. So acetylcholine receptor is a ligand gated cation channels, which is going to allow the exchange of cations. In some scenarios, there are ligand gated sodium ion channels, which are going to only selectively exchange sodium ions. They won't allow any other cation. Okay, so but in this case, the acetylcholine receptor is the ligand gated cation channel. It is not specific to certain cation. It can allow all of the it can allow the passage of all of the anions. Okay, and that reacts to the chemical stimulus. The chemical stimulus is here the ligand acetylcholine. Okay, so I hope this is clear. The third type of channel is mechanically gated channel, which reacts to the mechanical stimulus. So whenever there is some mechanical stimulus like touch, pressure, vibration, okay, all of those mechanical stimulus, they are going to exert some force on that channel, and that force is enough to distort its conformation. Okay, and this force is distorting the conformation, and that is opening the gate, and that will allow the exchange of the ions. Okay, then the last and very most important ion channel is the voltage gated ion channels, which are going to react to the change in the membrane potential. So due to certain reasons, let's say the membrane potential is changed. So let's say due to the leakage of ions, due to opening of leak channels, the membrane potential is changed from the 70 millivolt, it is going to minus 50 millivolt, and this change will be sensed by this channel, and this change is going to do some conformational change in the protein itself. So when the membrane potential is changing from minus 70 to minus 15, the conformation of the voltage gated potassium ion channel, here it is a potassium ion channel, it is changing. So here you can see this blob comes out from the channel, and this opens the gate, and that is why the potassium ions, they are moving from outside to the from the inside of the cell to the outside. So this is how the different ion channels which are residing on the membrane of the neuron, they are going to help in the maintaining the concentration gradient of so they are going to help in signaling. Okay, so we have discussed ion channels. Okay, so we will be discussing the signaling part, the organized signaling in nerve cell, how how upon the arrival of stimulus, certain ion channel opens, and then they change the membrane potential from resting membrane potential to some threshold level, and how the action potential is generated. We will learn about all those things in next lecture. Till then, see you. Bye bye.